Now, with the coronavirus protein synthesis, remember doesn't skip that. Or remember, coronavirus its genome is RNA. You go straight from RNA to protein. Now we finally get into mRNA vaccines. So how do you produce that? Well, what if we just take the mRNA protein, or not the protein, the mRNA that encodes the spike protein, and then inject that into cells? Remember, so what do cells do? They take mRNA and they make protein from that. So if you have RNA that encodes that spike protein, put it in our cells, make proteins, use our own ribosomes to make that protein. Now we can produce our own protein, spike proteins, or we can produce that spike protein in our own cells. Now, this is where a lot of misinformation is occurring because they're saying like, oh my God, this RNA is going to change our DNA. And the thing is that, does RNA go from RNA to DNA? Not in coronavirus, and we don't have the enzymes to do that in our own cells. mRNA is made all the time, but it doesn't go back to become DNA. It's a one-way street in human cells. mRNA, once it's used up and it eventually becomes degraded, and this is why they're working copies, and that's why if you have mRNA, it gets degraded and disintegrates over time. So remember that normally our cells present our own antigens and proteins inside our cells, and this is on those MHC class 1 receptors that pretty much every cell with a nucleus has. Now, what if one of these was a viral protein or spike protein? Again, if you have the mRNA from the spike protein, inject it into a cell and inject it into your system, and it ends up in a cell where it uses the ribosomes to make a spike protein. This spike protein can also end up on these MHC class 1 receptors. This can be presented to dendritic cells, T cells, B cells, and activate them and tell them like, hey, I found this protein and maybe you should mount an immune response to it. So that's how those mRNA vaccines work. And so what it does is pretty much skips that whole adenovirus, it skips the protein purification steps. Actually, I mean, I know it may seem like protein purification is really simple. If you work in a lab, it's really hard to even make what we call a vector to make a protein. And just making purified protein, that's why those, I think, purified protein vaccines, they're the last to come out because it's actually really hard to make any purified protein. And the cool thing about this mRNA vaccine, it's very easy to make mRNA compared to actually making an adenoviral vector. So it's like, this is why they came out first, and because it's very easy, like you can pretty much hire any sort of company that makes these what we call oligonucleotides, especially for PCR, polymerase chain reactions. Well, I think I'm getting too much into molecular biology, but it's very easy to make mRNA. That's what I'm getting at. So this is what's behind the Pfizer BioNTech, and it's not just the US, it's Pfizer, German company, and BioNTech. And this is approved in emergency use in 46 countries, or actually it should be more because it has the European Union. So yeah, this is the principle behind the mRNA vaccine. And then Moderna, it's not as proved as much because compared to Pfizer, remember Pfizer has a long history as a pharmaceutical company. It's the same Pfizer that made Viagra. Moderna is a relatively new startup compared to that, so that's maybe another reason why it's not as widespread. But they use the same general principle. Why they're very similar? They're probably using, if not the same, probably very almost identical genetic sequences between the spike protein. And this is why, if the spike protein mutates, this is why these will probably be the ones that come up with a new vaccine against that. All they have to do is take that mutation and encode it change the mRNA sequence that they inject. And now your cells are able to produce spike proteins that have that same structure as the mutated spike proteins in the variants. So that's why I think mRNA vaccines, it's kind of being proof of concept, and these are probably the ones that hopefully we don't have to deal with those variants for much longer, but the thing is that hopefully if there is like a variant that's resistant to the current vaccines, hopefully Pfizer and Moderna can change the sequence and to the new mutated variants, and then we can have a booster shot against those vaccines. And let's see, do I even have time? Okay, and let me finish with adjuvant. So yeah, those are the four main strategies behind the vaccines. We covered inactivated virus, we covered purified proteins, we covered adenovirus, and this is sometimes called just viral vectors, but adenoviruses is pretty much the other viral vector, and mRNA vaccines. And this is why they have different efficacies. They use different strategies. Again, the Chinese and Indian vaccines, they try to use the whole virus. The other three types try to use just to try to make a spike protein or expose your body to the spike protein. But how they do that, that's what the difference between the three. And I think what's the difference between Pfizer and Moderna? They're probably just whatever they use to actually get that mRNA into your cells. The efficiency of that is probably behind the efficiency with or not just like how the delivery method, but also like the timing is also different between Pfizer and Moderna. So it might be the ratio of the chemicals they use to, it's a process called transfection, but the way the chemicals they use to actually get mRNA into your cells and the ratio of that, and also the timing of when you're exposing your body is probably the reason behind the small differences between the Pfizer and Moderna. But the thing is that they have the genetic information, and just for the spike protein, that's why they're probably they're similar in their efficacy.